Welcome back to Pharmatech Frontiers. I'm Ritika, continuing our cutting edge series with Mohammad Aslam, M.Pharm from Amrita School of Pharmacy. Today we are exploring precision pharmacology. Mohammed, how is genomics revolutionizing our understanding of individual drug metabolism?
Hi Ritika. Actually, traditional medication were developed and prescribed based on how the average patient responds. So, clinical trials will determine a standard dose, and we will fix certain doses, and uh, we are administrating those to the patients. But in reality, people are an average. We all are like biologically unique. So that's where pharmacogenomics comes in. So pharmacogenomics, nothing, it's a study how our genes affect the way we respond to drugs. So some people might carry genetic variation like make a drug less effective or increase the risk of side effects, etc. So by identifying these markers through genetic testing, we can tailor treatment plans for individual rather than relying on trial and error. So this leads to the personalized medicine, so which is really the future of healthcare. It's not just about your symptoms; it's about your biology. We are now combining genomic with the data from your environment, lifestyle, even the microbiome to develop highly targeted therapies. So finally, we can say simply like we can ask like what drugs work for most people with this condition. The questions can become like what drug will work best for this specific person based on their unique genetic profiles. So it will be more precise, more proactive, and ultimately more effective approach to treatment.
Great, that was a very well rounded answer, I believe. So let's come into pharmacogenomics, and that its testing is becoming more and more sophisticated. So, what are the most clinically relevant genetic variants affecting drug metabolism today?
Pharmacogenomic testing has truly reshaped how we think about drug metabolism and individualized therapy. As we know, when we talk about drug metabolism, the main family is cytochrome P450 enzymes. So it's there actually main forms like a CYP2D6 to CYP2C19 and CYP2C9. So this polymorphism can determine whether the patient is poor metabolite or intermediate or ultra rapid metabolizer. So which directly impact the safety and effectiveness of widely used drugs like antidepressants, antiplatelet agents, and even warfarin, etc. So another uh, equally important are dihydropyrimidine dehydrogenase variants. So, especially 5-fluorouracils or pyrimidines, the drugs which comes under the class of pyrimidines, is metabolized by this DPYD enzyme. So, patient with a reduced function of DPYD alleles can experience severe, even life-threatening toxicity if standard doses are given. So, genotyping is now recommended before treatment. So mainly this CYP 450 family DPYD are recommended genetic testing before administering the drugs. Then another classic examples include thiopurine S-methyl transferase. So these are the enzymes which mainly metabolize drugs like azathioprine and mercaptopurine. So if these enzymes are not properly expressing in a human, there will be dangerous risk in bone marrow suppression, etc. So beyond this, we are seeing even guidelines are emerging for pharmacogens testing for UGT1A1 and SLCO1B1 with statin-induced myopathy risk. And rather than the other exciting thing is that organization like a Clinical Pharmacogenetic Implementation Consortium, that is CPIC, and Dutch Pharmacogenetic Working Group, DPWG, is actively publishing evidence-based clinical guidelines. So these tests are moving from the research bench to everyday clinical practice.
Right. So, how are polygenic risk scores being used to predict drug responses? And this seems much more complex to me than single gene testing. So, can you go into that and explain it for the audience?
Yeah, sure. Actually, traditional genomics often looks at single genes. For example, how variants in CYP2, C9 genes affect response to certain drugs. But in reality, we know many drug response aren't controlled by just one gene. Instead, they involve multiple genetic variants, each contributing a small effect. That's where the polygenic approach comes in. So in polygenic testing, looks at many variants across the genome at once. So by combining their effects into what's called a polygenic risk score (PRS), we can build a more accurate picture of how someone might respond to a drug. But there are certain challenges. We told about many genes are involved in metabolizing or to produce a proper effect of a drug. So uh, the challenges is that there may be thousands of variants involved, as it's not always clear which one matter most, how they interact. So this can be overcome by using machine learning. So machine learning makes a real difference. So this algorithm can sift through massive amount of genetic and clinical data, uncovering complex patterns that human might miss. And it's more sophisticated that simply adding variants up. Some variants may affect drug metabolism, other the drug's targets, or even some transport proteins. So machine learning can allow to weight each variant appropriately and account for interaction and interacting genetic information with other important factors like age, sex, and lifestyle. So this results in comprehensive genetic profile that better predicts how an individual will respond to medication.
Right. So, what about epigenetic factors and how do environmental influences on gene expression affects drug metabolism?
Uh, that's great point to bring up. Actually, if genetic is the blueprint, then epigenetic is the volume knob. It controls how strongly genes are turned on and off. So one common way this happens is through DNA methylation, where small chemical tags attach to DNA and reduce or even silence the activity of certain genes. So now when we talk about drug metabolizing enzyme like those in CYP450 family, epigenetics changes can significantly affect how well those enzymes function. For example, if methylation suppresses the gene that produces a key enzyme, a person might metabolize certain drugs more slowly, so which could lead to higher drug level in the blood or even more side effects will be there. So we know environmental factors will affect our genes like diet, smoking, pollution, even stress can influence these epigenetic marks. So two people with the same genetic makeup could respond very differently to drug if their environments have shaped their gene expression differently. Age also plays a crucial role. Like as we get older, our epigenome changes. So some drug metabolism genes may become more or less active, which is one reason why older adults often need different dosage than younger patients. So the therapeutic implications are big. We are starting to realize that understanding patients' epigenetic profile could one day help us to fine tune treatment, not just based on their genes, but also on how their lifestyle and environment have modified those genes over time.
Right. So we hear about multi-omics approaches all the time in drug metabolism. So how are researchers integrating genomics, proteomics, and metabolomics data?
Yeah, actually, the idea behind multiomics is to move beyond looking at just one layer of biology like genes, and instead connects the dots across genomics, proteomics, and metabolomics to get a full picture how drugs are processed in the body. So this can be achieved mainly by using system pharmacology. So it's a holistic approach that combines biological data, drug properties, and mathematical modeling to understand how drugs interact with complex biological networks rather than how a isolated gene or protein is interacting. So, for example, a system pharmacology model might start with person genetic variants. So, layer in protein expression data to see which enzymes are active, then add metabolomic data to track how the drug and its byproducts are behaving in the body. So, this can reveal subtle interaction that you would miss by looking at one layer alone. So one of the major goal actually is in multiomics is biomarker discovery. So by analyzing these different omics lab together, research can find much stronger and more reliable biomarkers. So these biomarkers might predict how well someone will respond to treatment, whether they will experience side effects or even what dose is optimal. And but there are certain challenges are there as we are dealing with a big set of data. And even we told it's not simply genes. We are also considering proteins, metabolite, etc. So the main computational challenges will be like massive and complex data, then different data types and formats. We know like genes will be in one format, then proteins and metabolic data will be in a different format. Then high noise and missing data are there. And another major challenges include model validation. So once you build a system pharmacology model, you have to test it rigorously across different population and settings. That take time, resources, and careful design.
Right. So, can you explain how machine learning algorithms, which are very much a hot topic actually, are being used to predict drug metabolism for multi-omics data?
Yeah, machine learning is becoming a power tool for predicting, like nowadays, how drugs are metabolized, especially when we bring in multiomics data like genomics, transcriptomics, proteomics, and metabolomics. So instead of relying on single markers, machine learning can capture the complex pattern across these different layers that influence ATME process like absorption, distribution, metabolism, and excretion. So, for example, a classical approach like random forests and gradient boosting are often used because they handle large, messy biological data sets quite well. So, more recently, deep learning models, including neural networks and even graph neural networks, can learn subtle nonlinear relationships. So, these models are particularly good at integrating molecular structure with multi-omics features to predict metabolic pathways and drug response. But big challenges is feature selection. So multiomics data sets often contain thousands of variable, but relatively few patients, which makes overfitting a real risk. So deal with this, researchers use dimensional reduction methods, autoencoders, and pathway-based filtering to keep the most meaningful features while minimizing noise. So finally, another thing is validation strategies. So that's also uh, like important. So nested cross validation prevent data leakage. Then external validation on uh, independent patient cohort ensure generalizability. And many group also combine model interpretability tools like sharp values, etc. So overall, machine learning, especially when combined with multiomics, is helping us move closer to truly personalized prediction of drug metabolism.
That makes a lot of sense. Thank you for the explanation. Now, moving on to digital biomarkers, which I think is present in abundance and are emerging as game changers. So, how might wearable devices help monitor drug metabolism in real time?
Yeah, We are in the era of where wearable and smartphone-based sensors are turning into body into a living laboratory, able to track how well medications are working moment to moment. So while we are talking about traditional pharmacokinetics relies on blood draws and occasional lab tests. So here in digital biomarkers give us continuous real world data. This is incredibly useful for understanding how patient is actually respond to a drug outside the clinic. For example, like continuous glucose monitoring and diabetic medicine. One of the best success stories diabetes management device like Dexcom or Libre continually track glucose levels via small sensors on the skin. Then another like for cardiac medications, wearable like Apple Watch, Whoop, or Aura Ring can track our heart rate variability continuously. Then latest some smartphone based monitorings are there even without wearable smartphones alone are becoming health tools. They can collect passive data like our from our movement patterns, sleep quality, voice and speech changes, then trying speed and touch screen behavior, etc. So there are like now even apps also can detect tremors, then cognitive slowing in a disease like Parkinson's, antidepressants. And even so, eventually, we may be able to correlate digital signals like skin temperature or movement with the real time drug concentration in the body using machine learning to translate wearable data into meaningful pharmacokinetic insight. So there are lot of techniques are emerging.
Okay, so what about liquid biopsies for therapeutic drug monitoring? How are circulating biomarkers being used?
Liquid biopsies are like emerging a powerful tool in therapeutic drug monitoring or TDM. So instead of relying on traditional blood draws just measure drug level, liquid biopsies analyze circulating biomarkers like cell-free DNA, RNA, proteins, and even drug metabolites from a simple blood sample. So, for example, in cancer treatment, we track tumor-derived cell-free DNA in blood to monitor how a patient is responding to targeted therapies. So, if resistance mutations show up, clinician can adjust the treatment before symptoms appears. That's real time and personalized decision making. So, so what exciting in this circulating markers gives us a dynamic and non-invasive insight into how the body is processing a drug and whether it's working. It's a huge step forward in making drug monitoring faster and safer and more personalized.
Right. So organ on chip platforms are revolutionizing drug testing, right? So how do these microphysiological systems model human drug metabolism?
Actually, organ-on-chip technology is actually a real game changer in drug development. So these are nothing microfluid devices about a size of a USB stick that contains living human cells arranged in 3D structures to mimic the function of actual human organs. So they are often refers to microphysiological system, and they give us an incredibly realistic view how drugs behave in human body. So one of the most widely used models is liver on a chip. Since liver is the main site of drug metabolism, researchers use this platform to study how drugs are broken down, how active metabolite form, and how toxic byproduct might impact liver cells. So these chips contain human hepatocytes, liver cells, along with the other support cells and even simulated blood flow, which allows the dynamic and functional drug metabolism studies in real time. And other than like We are seeing the development of multi-organ chips systems that link liver with the other organs like kidney, gut, heart, or even brain. This lets researchers to study how drug is absorbed, then metabolized, then filtered by kidney, and how metabolites affects the organ. So instead of studying one organ isolation, this platform simulate all body pharmacokinetics and even drug interactions. So now compared to traditional animal models, organ chips are some major benefits because they use human cells and the data is more predictive of human outcome, and real-time monitoring and toxicity, efficacy, etc. is possible. Then we know we can isolate the cells from patients, and we can try drugs metabolism in that one, which can be used as a personalized medicine. Prediction will be very useful, but it has some limitations. As we said, we are not quite at the finish line. These are systems are complex to build and standardize. There is variability between chips, and scaling them from high throughput drug screening is still challenges. Also, while they model many aspects of organ function, they can't fully replicate the immune system, hormonal regulation, and long-term dynamics for a chronic drug exposure level.
Okay. Thank you, Mohammed. That brings us to the end of the podcast, and thank you, listeners, for joining in.